Hello, everyone, and welcome to today's keynote webinar titled Role of LDL-Related Receptor Proteins in Viral Infection. Today's webinar is part of the 9th annual event in the Microbiology Week virtual event series. I'm Christy Jewell of LabRoots, and I'll be your moderator for today's event. Now, before we begin, I would like to remind everyone that today's event is interactive. We encourage you to first participate by communicating with other attendees using our live chat feature during the presentation. You can find that live chat located at the bottom left of your screen. You can also participate by submitting as many questions as you want at any time you want during the presentation. Just simply type them into the Ask a Question box and click Submit. We will answer as many questions as we have at the end of the presentation. Now, if you have any trouble seeing or hearing today's presentation, click on that Help Desk button from the lobby within the event. And finally, as a reminder, today's presentation is educational and offers free continuing education credits. Click on the continuing education credits image located at the right of your screen and follow the process to obtain your credits. Now let's get started. I would like to present today's speaker, Gaya Amarasinghe, Alumni Endowed Professor of Pathology and Immunology at the Division of Laboratory and Genomic Medicine, Department of Pathology and Immunology at Washington University School of Medicine in St. Louis. For a complete biography on our speaker, please find it on the left of your screen. And Gaya, welcome. You may now begin your presentation.
Great. Thank you, Christina, for the introduction. And I also want to welcome everyone, and also thank the organizers for the opportunity to speak to you today. So I will begin by talking to you a little bit about the work in my lab in our group at Washington University and with our collaborators. So we, as a group, are generally interested in studies of the host-pathogen interface. Particularly, we're interested in identifying gaps between host and viral interactions, as well as host and bacterial interactions. And in most of these studies, our goal is to understand molecular mechanisms that describe pathogenic events, but also some of these studies then lead to identifying therapeutic targets that can potentially be used as drug targets or interventions. So before I get into the topic today, which is to talk to you about LDL receptors and how these are LDL-related receptors and how these are used in viral entry, I want to just give you a little bit of a background of how we in our group think about this variety of different pathogens and variety of different host factors, and what are the unifying themes in terms of how we think about the science. So to do that, I'm just going to share with you a cartoon representation of a macromolecule. This is a molecule that has multiple atoms. In this case, I'm depicting a protein in the form of a cartoon here. And what you can see is that in this protein, there are multiple domains. This is a domain that is independently folded unit that has function. And what you see is that simply most of the studies over the last 50 to 60 years have shown that these molecules are not simply beads on a string, but rather arranged in complex interactions that allow or facilitate numerous domain-domain contacts. So what this leads to is these kinds of contacts leads to regulation and control of function. For example, here, this contact between the blue and the yellow module eventually allows for the yellow module, if it has activity or enzymatic activity, it to be repressed in this state and then activated in this state. So, so what do we do in the lab? So in each of our projects, we think about two very simple questions. We ask, what are the physical properties that gives rise to these complex behaviors and regulation? And the second question we ask is, how are these properties changed by others? In the first example, in terms of the properties that gives rise to these complex behaviors, what we think about is structure, kinetics, that means how fast do things happen, and then thermodynamics, or how likely is an event going to happen. And then the second question, this is really the question related to pathogenesis and pathology, and that is, how do these properties, the structure, the kinetics, the dynamics, how do those change when they interact with other molecules? In our case, oftentimes that other molecule is a viral or a bacterial protein. So as the topic, the title suggested, we're going to talk to you about, I'm going to talk to you about LDL-related proteins, and this is a project that's a very close collaboration between my group, a number of my colleagues are shown here, as well as Amy Hartman's group at Pittsburgh, Daisy Liang, Takeshi Gawa, Shasha Q, and Michael Gross at Washington University. And this work really began about, at this point, four plus years ago, by a very talented postdoctoral fellow in my group, Safta Ganai. But he was, through this journey, he was equally helped by a number of individuals, including Dave Price, Fahim Fatma, Griffin Brown, Wenji Wang, Annie Fang, and Nicole Wagner. And these individuals worked as a team at WashU, together with our colleagues in Pittsburgh, to bring you the data that I'm going to share with you today. So let's take a step back and think about viruses. Viruses have different forms and different genetic material. And I think given the last few years, I think everybody now fully appreciates the potential both socioeconomic and public health risks that pathogens can form in part due to the COVID-19 pandemic. And so in this case, what I'm showing here is the Baltimore classification. This is the classification that groups viruses into double-stranded DNA, single-stranded DNA, single-stranded RNA in the negative sense polarity, single-stranded RNA in the positive sense polarity, double-stranded RNA, and then, of course, you have retroviruses, the genomic materials, RNA, but they can convert into a DNA and then followed by reverting back into DNA. And these are basically the 6 groups in the Baltimore classification. And so today, my group historically has been has conducted a lot of work in filoviruses, but I'm not going to talk to you, and paramyxoviruses, and I'm not going to talk to you about those 2 families, but instead I'm going to share with you really the work from the last 4+ years between my group, Daisy Liang's group, and Amy Hartman's group, where we've been able to really dissect various aspects of the Bunya virus biology. So what are Bunya viruses? This is a very large family of single-stranded negative sense RNA viral viruses. There are about 300 members in this family, and therefore they're grouped into 5 different genera. And what's interesting about these viruses is they're transmitted through insects, arthropods, or rodents, and in some instances, most of these viruses are transmitted, and very rarely do humans get infected, but during human infections, they lead to significant, potentially significant outbreaks. So the genome of these viruses can be multi-segmented. In the case of Bunya viruses, they're either bi or tripartite. And occasionally, some of these viruses have the ability to cause severe hemorrhagic fever. And again, given our previous work in filoviruses, this seems to be a theme in the lab. We are interested in understanding how do pathogens, infections or pathogenic infections lead to severe hemorrhagic fever. So the particular virus that I'm going to focus on for much of my talk today is a virus called the Rift Valley fever virus. And you'll see this acronym RVFV. And this is a zoonotic pathogen. What I mean by that is typically it undergoes what we call an enzootic cycle within the vector host. And occasionally you have an epizootic or an epidemic cycle where the vector host is able to transmit the virus to a different host, in this case, ruminants, sheep and cows and goats. And then occasionally you have incidental spillover from that into humans. And this is when you have outbreaks and pandemics and eventually epidemics, and eventually pandemics, that means into multiple locations. Typically, Rift Valley fever virus is infections are seen in sub-Saharan Africa, and this is a CDC outline where you can see the distribution map of the virus. So the viral disease outcomes are different for livestock and humans. In acute disease, In the case of livestock, you see typical fever, weakness, anorexia, diarrhea, and nasal discharge. There's an age-dependent mortality. The younger animals tend to see more significant mortality rates, up to 90%, whereas adult animals see much less mortality rates compared to the young animals, in this case, 30 to 40%. One of the most significant outcomes is you see fetal spontaneous abortions in infected animals, and this not only impacts the livestock, but also prevents the subsequent generations from being born, and therefore there's a big, not only an impact on the immediate impact, but also potential long-term socioeconomic impact in affected areas. In the case of people, this presents as a non-lethal febrile illness. Occasionally, severe cases lead to hepatic, i.e. liver disease or hemorrhagic disease. And occasionally, we also see encephalitis, infection of the central nervous system and the brain, as well as ocular infections. Again, these are some of the areas that are immune privileged sites. And so it's important to note that in severe cases, that these can lead to significant damage into organs that are very difficult to protect. So we began this study, we initiated this in 2018, I think, using a screen or a study that I think a lot of groups have used to identify specific host factors for a variety of events. The idea here is to use the CRISPR-Cas9 system. The idea, we take a set of cells that express the Cas9 protein, and by transducing the single-stranded guides, we can selectively remove a given protein in a given cell. This then leads to a clonal population where each of the non-essential genes in the genome have been removed, and then we can apply a selection pressure. This can be infection with a virus in our case, and then ask, what is your population at the beginning? What is the population at the end? And this difference, the delta, gives us the criteria for survival. And we can also look at the positive selection and the negative selection. And this gives us key genes that are important for this process. And of course, this part is difficult to do, but one of the most significant aspects is the validation. This is very, very hard and very difficult. And that's what really the topic that I want to share with you. So as I mentioned at the outset, this is a very close collaboration with Dr. Hartman's group at the University of Pittsburgh. And this is Safta Ganai, who's the postdoc in my group, who actually at the beginning of the project from both labs spearheaded this work and actually got this to move to the point where we were all able to sort of begin to interpret some of the results. So what I'm showing you here is an image of cells. What you can see is that upon infection, immediately, this is a highly cytopathic virus. Most of the cells are depleted. These are dead. Cells are removed. And you can see after day 8 and day 13, you can start to see clusters of cells or clumps of cells appear. These are clonal populations of survivors. And if you take this clump of cells and replate them and reinfect them, what you can see is that we see significant resistance to infection. This allowed us to say that whatever the protein that is missing from these cells are now a key factor to provide resistance or reduced infection. So these cells, what I didn't mention was at the beginning was that the CRISPR-Cas screen is barcoded. So we can ask what gene is missing in these cells. When we did that, what we were able to see was that LDL receptor family protein, LRP1, along with number of proteins that are in the pathway that helps produce LRP1, were also missing in this system. And that was what we were able to, that is what Safta was able to glean from this data. So here's LRP1. I'm depicting that in this green bar. And you can see that RAP, receptor-associated protein, it's something that keeps these LDL receptors ligand-free through the ER-Golgi network. And also, a HSP90 family member, GRP94, was also present, and you can see that it is important as a chaperone for folding of LRP1, and these are all important proteins that are in this pathway. So identifying these actually provided us the basis to understand that, in fact, we may be onto something that each of these proteins may be playing a role in terms of enabling Rift Valley fever virus infection. So as I said, the screening part is difficult to do, but not the most difficult thing. That is the second most difficult thing, because the most difficult part is actually the validation. So to do this, we enlisted help from our genome editing center, Shao Shakir, and Mike White, who runs the microinjection and the mouse breeding facility. And one of the first things we did was we generated clonal knockout cells. What you can see is a clonal knockout cell and an allelic knockout cell. And when SAFTA was able to infect using Rift Valley fever MP12, this is a vaccine strain of the virus that also encodes sort of GFP molecule. You can see that the wild type cells can be readily infected, but then when you take this R4 cell line, which is the cell line that is lacking the expression of LRP1, you can see there's a clear reduction in infection. When we remove genes, you can do damage to the cells themselves. So one of the things we did was to ask, can we infect other viruses such as influenza or RSV, other RNA viruses, and ask, how do these perform? And you can see that there's no significant difference between the wild type and these knockout cells, suggesting that we didn't really completely ruin the cells to a point that there's significant resistance in the knockout cells, but rather there's a specific resistance to infection by Rift Valley fever virus. This again provided us with the basis to move forward to ask, why do we see this resistance? So to do that, SAFTA, Annie Fang and others in the lab did several experiments. So here's LRP1. I'll get into a little bit more details later on. It is a fairly large protein of 515 kilodaltons in the ectodomain and another 75 to 80 kilodalton in the transmembrane and cytoplasmic region. So this is a very, very large protein. So what they did was first read the literature. And they realized that we were not the first ones to identify and study LRP1. In fact, there are thousands of papers that cite LRP1 as an important molecule for various processes, particularly given its significant role in LDL binding and clearance. So the literature had used a number of different ways to overcome the large proteins in terms of expressing them ectopically. One of them was to make these mini constructs where the transmembrane and the cytoplasmic region is identical, except now you have cluster 1, cluster 2, cluster 3, and cluster 4. Each of these clusters can be then tethered to or fused to the same transmembrane region and the cytoplasmic region. And when we did this and trans-complemented our original R4 knockout cell line, what we see is we can recapture activity for cluster 2 and cluster 4, but not as much as the wild type cells, suggesting that these can partially restore infectivity in these cells. We then did another experiment, and that was to generate a series of soluble constructs of clusters 2, 3, and 4, and then using the fragment crystallizable or the FC region of an antibody fused, and these soluble receptors were able to again show that they can bind to the Rift Valley fever virus GN protein, because Rift Valley fever virus has two glycoproteins, GN and GC, and we had ruled out GC in the data for which the data I'm not showing here, but you can show clearly that the in a pull-down assay or an immunoprecipitation assay, you can show that here's the glycoprotein GN and here are the different clusters, and you can see cluster 2 and cluster 4 clearly interact with each other. So now we have shown that some of these clusters are sufficient for infection and transcomplementation, and we've shown that they can bind and interact. So Wenji Wang began this studies when he was a postdoc in my group. He's now managing his own group in a pharmaceutical company elsewhere, together with David Price and Fahim Fatma in our groups, was able to tackle this question and ask a very specific question. Why does Rift Valley fever virus GN bind, and can we block this binding? As I mentioned earlier, we are not the first ones to study this system, so we were able to identify previous ligands. One of them is this receptor-associated protein, which also turned out to be a hit on our screen, And we were able to look at this and say, how does this RAP protein interact, and can this RAP protein block Rift Valley fever virus GN? To do this, again, we went to the literature. There were mouse MRAP, that's the acronym MRAP, receptor-associated protein, domain 3. And there was also a known mutation of domain 3, which reduces the binding. And when we used these two reagents, the MRAP D3 and the mutant MRAP, what we were able to first do was to perform a binding assay. This is a biolayer interferometry. You can see the increase in this signal shows binding, And in this case, when you use the mutant version, you can see that it has significantly diminished binding. And this protein binds to cluster 2 and cluster 4, again, consistent with what he had seen before with GN binding, and therefore provided us the initial test to ask, can then RAP D3 inhibit Rift Valley fever GN binding? We first performed a biochemical assay. Here's the Rift Valley fever GN binding. Without MRAP D3, it binds very well, but when we dose-dependently increase the MRAP D3, you see significant decrease in binding, suggesting again that these two proteins may bind in the similar site and therefore compete each other. Safter then took this information and performed what I think is a very critical experiment, and that is he increased the concentration and asked, just because biochemically we block this, can we now block this in infection in cell culture? And as you can see, the EC50 of less than 1 microgram is what we see. Here's the quantitation of the data. Here's the raw data. No infection, you get infection. And as we increase the MRAP, you see significantly reduced infection, eventually reducing it better than 90%, suggesting that, again, not only MRAP D3 can bind Rift Valley, the LRP1 in similar places as Rift Valley fever GN, but it can block infection. So one of the biggest challenges in identifying receptors or entry factors for viruses is that a lot of times they tend to be tissue culture artifacts of things that you see in one cell line but not reproducible in the other. So with this reagent, MRAP D3, we asked, can we go into a series of taxonomically distinct cells, cells derived from taxonomically distinct organisms, and ask, can MRAP D3 inhibit this? There were two reasons why this experiment worked. One, we had high quality MRAP D3 in the lab. Second, we also were able to use the fact that LRP1 is fairly well conserved across different taxonomically distinct organisms. So using 5 times the LD50 or the EC50, we were able to use 5 micrograms, and each time in humans, bovine, hamster, and monkey cells, in each time you can see a significant decrease of infection upon MRAP treatment. We also checked different cells derived from different tissues, in this case a hepatic cell line and a neuronal cell line, and in each instance we see significant decrease in infection upon MRAP treatment, again suggesting that, one, these MRAP from mouse MRAP binds the LRP1, and the second thing is that we can actually prevent infection from that. So next we asked, it's nice that we can do this in vitro, could we do this in animals? And in this case, we, Amy Hartman's group performed an incredibly brilliant experiment, and in this, they asked the question if mice treated with MRAP prevent Rift Valley fever virus infection during intracranial infection. This work was performed by a now former graduate student in the Hartman lab, Madeline Shores, and what you can see is that the red bars here is a 10 PFU, 10 plaque forming units, and mind you, the LD50 or the lethal dose is less than 1 plaque forming unit. And you can see that the mice succumb to the virus between day 2 to 6. But if you now have MRAP D3 in this intracranial infections, you see significant survival compared to this, right? So the second thing they'll note is that if you use the mutant version of MRAP, we get significant death and no survival. And very similarly, if you use a control protein, you end up with a near similar result. What we see here is significant survival and also increased significant survival and increased time to death, right? So these suggested to us that, in fact, MRAP and LRP1, the Rift Valley fever virus GN may be binding in the same place, and this could be giving us some information and insights into the entry mechanism. This is just the quantitation of the viral RNA by qPCR. You can see again that this column is the MRAP-treated animals. They're very different, the shades of red you can see. And of course, very similar data is obtained in the plaque assays. These are two ways of quantifying the viral load in the liver and the brain. So here's a little bit of immunohistochemistry. Red is the color depicting the viral infection. In the untreated animals, you can see significant virus in the liver. This is a highly hepatotropic virus, and the brain. And you can see again neuronal and encephalitic disease. And in the case of MRAP treatment, you can see very, very little color. And of course, for interest of time, I'm not going to show you the controls, but we've done primary delete experiments and shown that this is not an artifact. This is in fact actually virus missing. And we see in the mutant infected animals, you see some infection, not as significant as the wild type, right? So this provided us with a basis suggesting that LRP1 is a critical entry factor. We then asked the question, is this specific to Rift Valley fever virus? That's this here. Or other Borneo viruses are also responsible similarly, Oropouche, lacrosse, and what we see is that indeed they show significant usage of LRP1, but not Zika, not SARS, and not measles. This again provided us with the basis for wanting to pursue this further to ask, is this a potential pan-Borneo virus entry factor? And what you can see is we can use different cell lines. This is an lung-derived cell line. This is a HEK293 T cells. And we've done in multiple cell lines. And again, we see very, very similar results. And just like we saw with the Rift Valley fever virus, I'm just sharing with you one data set from the study we published last year, where you can see that just like Rift Valley fever virus, if you're during an orthoborneo virus or a push infection, you can see very, very similar survival upon treatment of MRAP-D3 compared to untreated or mutant-treated animals. Again, this is suggesting that we have been able to identify something that is important for infection, and blocking that protein limits infection and increases survival. So to further dissect the system and to ask tissue specifically, how does LRP1 facilitate or promote infection? Safta and Maddie began a series of studies where they used a new mouse line that we generated here at Washington University in St. Louis, where we specifically knocked in developed a fluxed mouse. This is basically inserting LOXP sites in between the open reading frame or the coding region of the LRP1. And so with Cre recombinase, we can then remove it, remove the expression of the gene. And this is what Safta is showing here in a Western blot. This is mouse embryonic fibroblast cells. These are cells that are derived from mouse embryos. And what you can see is that when he add Cre treats or retro Cre treats, you can see that significant reduction in LRP1. And correspondingly, when he takes these cells, whether they're the flux animals or the wild type animals, you can see the wild type animals can support infection, but the fluxed animals treated with Cre recombinase has significantly reduced infection. And again, in the interest of time, I'm not showing you the data here, but when we subject these same cells to control infections such as influenza or RSV, we see very, very similar results. So next we ask, what happens when you specifically remove LRP1 in the liver? Because Rift Valley fever virus is a known hepatotropic virus and causes significant damage to the liver. So given the data that I've shown you here, removal of LRP1 significantly impairs the ability of Rift Valley fever virus to infect you would imagine that you'd get a survival curve that is similar to this curve. And unfortunately, or fortunately, that is not what we saw. So here's the floxed animals. These are the ones that are still expressing LRP1. And here's the floxed animals crossed to albumin Cre, which is expressing Cre recombinase just in the liver. What we saw was not survival curve like this, but instead what we saw was actually an increased time to death. The mean survival date went from 4 days to about 9 days, right? So this was puzzling, in some ways disappointing, but nonetheless, undeterred by these results, we went ahead and asked, can we characterize these mice and ask, where is this extended time to death coming from? And can we better understand how this result relates to the disease progression in infected animals and ultimately in humans? So to do that, we then looked at the pathology of the different tissue. In this case, I'm just sharing you data for the liver, and what you can see is an uninfected liver looks really nice in terms of the color and its presentation. Shiny, purple, pink, reddish color. And what you can see is in the albumin Cre liver, you can see 5 days post-infection, it's much more similar to a healthy, untreated liver compared to the liver derived from a mouse that succumbed to the virus, day 5 sac mouse, or a mouse that succumbed to the virus on day 5. So this immediately told us that the removal of LRP1 reduces or significantly diminishes, and this was further confirmed by immunohistochemistry studies where you can see that the wild-type mouse or the floxed mouse has significant infection and succumbed to the virus by day 6 to 7, whereas the ALB-Cre mice have very little infection in the liver in day 5, similar to day 7 and day 14. And these survive, but they ultimately still succumb to the virus, and therefore we surmise from this information that this is unlikely to be a hepatotropic or hepatic cause of disease, but rather some other systemic infection. So it wasn't entirely surprising when we then subsequently looked at the brain. What you can see is in the LRP1 flox mice, you see day 5 and day 7, you see very little virus highlighted by this pink color or the magenta color. And whereas the ALB-Cre treated animals, even beginning in day 5, we start seeing signs that the virus has infiltrated the CNS, in this case, the brain tissue. So what that suggested to us was, instead of it being a systemic or a peripheral infection, it has now gone into the CNS and the brain. This is also interesting when we then went back for each animal and compared days post-infection, the number of mice versus the neurological endpoints. We saw a very small fraction of neurological endpoints for the mice that were in our control group. These are the floxed animals, whereas we saw a significantly higher number of neurological signs at the endpoint for the LRP1 floxed mice crossed to the ALB-Cre. So that means these are mice that are lacking LRP1 in the liver. So we further went ahead, and this is again work from Mary Schultz in the Hartman group. And what she did was, each mouse, depending on the time of death or the day of death or euthanasia, you can see when she looked at the liver titers for the viruses, and each point here I note is a single animal. The floxed animals, not crossed to ALB-Cre, that means they have functional LRP1 in the liver. Most of those animals show high viremia, but they succumb to the virus very early. These open squares are the animals that are missing LRP1 in the liver, and you can see that these animals actually have significantly reduced viral loads in the liver, but in contrast, their brain viral loads are significantly higher, again suggesting that the virus is able to infiltrate the CNS without additional replication and propagation within the liver. So I want to, before I go into describing the more global impact of LRP1, I just want to very quickly summarize what I've talked to you about the for the last 30 minutes or so. And that is that through a series of studies that we conducted with our collaborators, we've been able to identify LRP1 as a critical host factor that promotes infection of Rift Valley fever virus entry. And I hope I convinced you with a couple of slides of Oropouche and other viruses that this may also be important for multiple bunyaviral entry and infection, and support multiple bunyavirus entry and infection. And of course, I hope that I was able to convince you that blocking the LRP1 binding by the viral glycoprotein then significantly limits infection in an in vivo model, and this also increases survival. And correspondingly, I showed that tissue-specific removal, in this case, I shared with you data for the liver. We have ongoing studies in other tissue, and you can see that this increases the time to death. And also, by increasing time to death, then we observed that there's potential CNS disease. And of course, we are very interested in sort of following up on these studies to identify how does this protecting in the liver then lead to significant CNS disease. So I now want to sort of switch gears a little bit and talk a little bit about future directions and tell you about why we think that LDL-related proteins are most significant. It turns out LRP1 is important for tau internalization, toxin entry, and cholesterol homeostasis. So there are lots and lots of interesting paths forward for us and others. So in the remaining 5 to 6 minutes or so, what I want to do is to share with you some thoughts that have come about, because turns out we were not the first ones, or the only ones studying LDL receptors. There are a number of groups, including my colleague Mike Diamond and others down the hall here at Washington University School of Medicine in St. Louis, as well as Jonathan Abrams at the Harvard Medical School, and Yi Zhang and the National Academy of Sciences in China, in one of the national laboratories, have each identified important interactions between LDL family proteins and other viral infections. In this case, what I'm sharing with you are data for multiple alpha viruses, some looking for resistant alpha virus, and also Venezuelan equinoencephalitis. These are the New World alpha virus and some Old World alpha viruses, and suggesting that, in fact, multiple viral families, now not just Bunya viruses, also potentially uses LDL family receptors. So what do the data looks like? I'm not going to belabor this. These are some of the citations, so you can look them up. But what's interesting to note is here's LRP1. It has each of these green bubbles is a ligand binding domain. They're called La repeats, or CDRs, so cysteine-rich domains. I will refer to them as CDRs throughout the rest of my talk. It also has beta propeller domains, EGF. These are epidermal growth factor repeats. There's number of glycosylations, and importantly, there's this NPXY motif. This is important for endocytosis and endocytic processes. And what you'll see is that here's LRP1. It's fairly large. It has 34 of these repeats. We've used this little rad, or LRAD3. It has 3 CDRs, and VLDR, a very, very low density lipid receptor, and ApoER3. This is also called LRP8, and these each have 7 and 8 LA repeats, or the CDRs. So then a question arises, how do these different ligand binding domains interact with the glycosylation? And I had already shared with you data showing that if you take any of the mini clusters, the cluster 2 and cluster 4, which each has 6 to 10 domains, can each promote infection, but not to the same level as the full-length protein. This gives us 2 sort of very simplistic options. One is that the multiple binding sites creates avidity, or multi-binding events, which prolongs the half-life of an interaction. And the second event is that potentially there's a ping-pong effect between these different ligand binding domains of the CDRs, and that then increases the half-life or the attachment on the cell surface that leads to higher levels of infection. I'll give you the short answer. We don't know the answer yet, but I think we have some clues. So the first clue comes from this study from Yi Jiang and et al., where they looked at the semligiforous virus binding in complex with a very low density lipid receptor, VLDR. And in the structure, which they used one of the ligand binding domains of the repeats, what they can see is that in fact it binds to the domain 3, so the E protein, E1 and E2 are the two glycoproteins that are equivalent to Rift Valley fever viruses GN and GC, and you can see that it specifically binds to domain 3 in this case, and it's able to prevent potentially attachment of the virus to the cell surface. So this then led us, together with some of the published data, to propose the following model, in that if you have a small number of binding events, you have low affinity and potentially low rates of infection without secondary attachment at another site. But if you have multiple binding sites, you have multiple domains, then you have high rates of entry levels, longer half-lives at the surface, and therefore correspondingly high rates of infection. So with this, I want to come back to the original model, the cartoon representation that I started with, by telling you that multi-domain proteins like LDL family receptors have many, many complex interactions. And I hope that I was able to provide some data to convince you that genome-wide screens are very important for understanding either gain of function or loss of function. And I hope I was able to convince you that LRP1 is an important attachment and entry factor that supports Rift Valley fever and other bunioviruses into cells derived from taxonomically distinct organisms. And the second aspect, I think this is more general and more important for understanding biology, and that is that modularity that is present within this LDL receptor family provides a really broad specificity, and this is a key theme in my group and many others, where we are trying to understand how the host-pathogen interface is able to differentiate self from non-self, and this ability to have broad specificity is important for early activity by cells to identify foreign pathogens, and hopefully, as we begin to continue to delve into the different aspects of this engagement with LDL receptors, we will be able to better understand the role of broad specificity in infection and immune response. So with that, I want to stop and thank a number of people. I think I mentioned Dr. Hartman's group along the way. Amy has been an incredible collaborator over the last 5 or 6 years. Madeline Schultz, who's since graduated, did bulk of the work that I described with animal studies. Cynthia and Rachel have joined, and as well as Zach, who's recently joined the group and working with us. And I'm incredibly grateful for Amy and her group. Here at Washington University, we have a really vibrant microbial infection and immunology community. I specifically want to thank my colleagues, Sean Whalen and his group, Mike Diamond, his group, Anthony Overdall, as well as our colleagues in Kentucky, Becky Dutch and her group, for some of the important insights and reagents and support. I didn't get a chance due to the topic and timing to describe a lot of our filovirus work, but again, this is where we began, particularly Chris Bassler, Rob Davey, Tom Geisbert, and Alex Bookrev. I'm really grateful for their role in sort of getting us to think about host pathogens and work in this area, and for some of the ongoing work. In my final slide, I want to acknowledge my group, a number of members whose work I highlighted, for making it actually exciting and interesting to come to work every day. I also want to thank my colleague Daisy Liang and her group. Both of our groups worked very closely together in many of these studies. And here's actually a recent picture of the Hartman group together with a number of groups in the Liang and Amarasinghe labs. And you can see that we actually are highly interactive, even though we are separated. One group is in St. Louis and the other one is in Pittsburgh. And before I end, I want to thank a number of the funding agencies who continue to support, including Washington University, NIH, and DOD. And again, I want to always mention that we are looking for trainees who are interested in joining us. So I will thank you for your attention, and I'm delighted to have this opportunity to speak to you, and look forward to taking any questions you may have.
Gaya, thank you so much for your informative presentation. We will now start the live Q&A portion of our webinar. Now, to our audience, if you have any questions you would like to ask, please do so now. Just submit them in the Ask a Question box, and we will answer as many of your questions as we have time for. Okay, let's get started. Now, Gaya, we have quite a few questions that have already come in, so I'm just going to dive in. Let's start with this one. LDL and lipids are important for normal function. So how does viral infection using these receptors impact cells and therefore animals?
Okay, yeah, so Christina, this is a great question, because this is an important one for both normal function and in the case of infection, because these are receptors that have really a day job, so to speak, and their job is not necessarily to help viruses get into our cells. And so I think the short answer is we still don't know how much impact it has in terms of the membrane fluidity, how the membrane would act before versus after infection. Because what we still don't know is these are receptors that are highly regulated, and they're recycled repeatedly from the cell surface back through the recycling endosomes, and they come back to the surface. So these are very, very well orchestrated and highly controlled process. However, if I were to sort of take a guess or to speculate, I think it will probably have a significant impact on the cell's ability to respond to external stimuli, because you now have something that is normally present to receive and process and capture lipids, LDL, low-density lipoproteins, and now you are using it for viruses, and we don't yet know what is the fate of this receptor. So I think the speculation is that this particular aspect of engaging may have more than just the effect of allowing a virus to come into the cell. So that's an important question. And so this is something that we are continuing to think about and to identify ways in which to address it, to better define what the ultimate impact is. And also a related question is whether those change from cell type to cell type. And this is important because cells like dendritic cells and macrophages are like first places that gets infected. And we want to really understand how that affects, and therefore our ability to fight not just Borrelia viruses, but other viruses, and whether that actually limits our ability. So great question, yeah.
Thank you, an excellent explanation, Gaya. Let's grab our next question here. Most surface proteins are glycosylated. Does glycosylation impact this interaction or your overall observations?
Yeah, so thank you, Christina. So that's an interesting question in that a lot of surface proteins, as the question notes, are decorated with different forms of sugars or glycosylations. And this helps to signal to ligands that these are the ligands to bind, or prevent them from degradation and increase in the half-life. So one of the biggest questions we had when we began this study was, is the Rift Valley Fjord glycoprotein binding to LRP1 dependent on the glycosylation of either of the two molecules, because they are both glycosylated? So one of the very first things we did, and this is buried in the supplementary data in one of the papers we published initially, showing that these two proteins are important, in that the interaction itself is not glycosylation dependent. So we can remove all the glycans through various mechanisms using deglycosylases or hydrolases or growing cells in cells that cannot glycosylate proteins and generating material that way. We have shown, we went into excruciating pain and sort of effort to show that that this binding interaction biochemically is glycosylation independent. So that's part one of the answer. And the part two is that even though they're not necessary for glycosylation for binding, it is likely that both of these proteins are glycosylated in an infected setting. So therefore, they're going to play a role. And I think this is a really open question for us to address, is what is the role of glycosylation? Does it make it more infectable on either part, or less infectable? Or does it give specificity in that now it allows the virus to hone in on certain cells that are glycosylated on LRP1 in a certain way. So these are all very, very interesting and important questions moving forward, but open for sure. Yeah.
Very good. And Gaya, let's go with this question, and I want to make sure I'm saying these abbreviations correctly. By design, MOABs to PCSK9 that are used therapeutically to treat hyperlipidemia increase LRP1 expression on cell surfaces. Might this increase vulnerability to RVF infection?
Yeah, no, that's a great question. So the question that the person is asking is the second slide that I showed, or the third slide that I showed, where I shared with you that the way we identified LRP1 is by this barcoded screen, and that then led to identifying not just LRP1, but then RAP, receptor-associated protein. And I also had PCSK9, but PCSK9 removal actually led to more infection, not less infection. Okay? So the person is absolutely right that removal of PCSK9, this is an enzyme that removes LDL family receptors from the surface, then leads to more infection. And the MABs, the monoclonal antibody therapy for hypercholesterolemia, these are people who are resistant to statin treatment or non-responsive to statin treatment, but are responsive to blocking PCSK9. There's a genetic mutation. And so the question is then, are you going to be sensitive then to infections? And the hypothetical answer is most likely. But at a cellular level, we can do these experiments where we put in PCSK9 or overexpress PCSK9, and that's exactly what you see. And the screen results actually are consistent with the observation. So I forgot whether there was a second part to that question in terms of asking, is there a population, is there significant danger then to taking these monoclonal antibodies? And my answer to that is obviously any medical decision needs to be made in consultation with their physicians. But that said, I think the parts of the world that these viruses are available and the vector hosts are in a limited geographic distribution. So I think we are safe to take some of those antibody treatments, and and the liability is very, very small, I would say. Yeah.
Thank you, Gaya. And it looks like we have time for one more question. LRP1 is a scavenger receptor, but your data shows some specificity, but unclear where that is coming from. Do you care to comment on that?
Yeah. So since we have 1 minute, I'll give a short answer that we don't know yet, but we are looking into that. And I think the model that I presented at the end, which is this multi-site binding, probably gives longer half-lives on the surface of the cell. So that's probably a hand-waving way of saying we don't know yet, but longer a virus stays on a cell, more likely it is to get in through processes such as macropinocytosis and endocytosis. So other processes may be helping or working in concert with that binding, and that multi-site binding may keep a virus long enough for other things to catch on. Yeah.
Gaya, thank you for this incredible Q&A session. So informative. Do you have any final comments for our audience?
No, I just want to thank everyone for their questions and interest. And I, again, want to sort of end on thanking my group and Daisy's group and Amy's group. I think all of them are shown here, for making this science and all the other stuff we do such an exciting and interesting thing, and for their dedication and hard work. So I want to end there. And again, thank you for your attention and opportunity.
Thank you again to Gaya Amarasinghe for his time today and his important research. Before we go, I do want to thank our audience also for joining us today and for their interesting questions. Now, those questions that we were unable to answer today and those that are submitted during the on-demand period, they will be addressed by our speaker via the contact information you provided at the time of registration. Today's webcast can be viewed on demand. for 2 years, and that goes through September 17th, 2025. LabRoots will alert you via email when it's available for replay, so watch out for that email. We encourage you to share that email with your colleagues who may have missed today's live event. That's all for now. Until next time, have a great day everybody. Bye-bye.